What is this cell?
Which one?
T
First,
cells.
T cell.
Killer T cells. No.
Cytotoxic.
yeah, cytotoxic T cell. And what is its function?
Kill viruses.
Kill virus infected cells or tumor cells, any parts. So, next one is T helper, because you see the CD4 molecule. What is its function? They help, and they help by giving some money. They help through the cytokines, and those cytokines that are secreted by these cells bind to other cells and activate them to do their functions. And looking at the nucleus on this cell, what do you think it is?
It looks like a neutrophil.
Coming closer, because it's round in shape, I would expect you to tell it's a monocyte. A macrophage will have different shapes because it's in the tissues. It will still have a kidney-shaped nucleus. Okay, last class we were talking about dendritic cells. Dendritic cells are one of the antigen-presenting cells. But coming here, you see all the three cells that we call them as antigen-presenting cells. Again, repeating, the T lymphocytes do not recognize antigen when it enters the body, but it's the antigen-presenting cells, such as these, that take up phagocytose it, break it down into small peptides, and then present it to the T cells. So these are antigen-presenting cells, dendritic cells, macrophages, and B lymphocytes. Where they are exactly present is shown here. Don't have to say much about that. Macrophages, peripheral blood, organ surfaces. But importantly, B lymphocytes, although it says peripheral blood, that's a small number in the peripheral blood. It's mostly T lymphocytes that are present. Okay? But they're mainly present in the lymphoid tissues that we will be talking in this class. Okay? Lymphoid organs like lymph nodes, spleen, Peyer's patches, and all those. Okay, we said antigen processing or antigen presentation. These three cells present antigen to the T lymphocytes. But what exactly is happening? Say this is an antigen presenting cell, such as a macrophage. Think that the antigen, it could be a virus or bacteria or anything, is phagocytosed, broken down into small peptides, and then these peptides are presented on the surface of this antigen-presenting cell in conjunction with this MHC molecule. So these are in turn recognized by the T lymphocyte with its T cell receptor, which we'll talk about. So that's antigen presentation. Again, going through these T cells like this one do not recognize this antigen by themselves. It's first taken up by these antigen presenting cells, and then a small peptide is presented on their surface in conjunction with MHC molecules. This we will talk again later, but then this small peptide will then be recognized by the T helper cell, and once it recognizes that small peptide, it will secrete those cytokines, which will then bind to other immune cells and activate them. Okay? Am I clear on that antigen presentation? skip this slide, so ignore that slide for some time. We'll talk about this in later classes. Going on to the polymorphonuclear leukocytes, or the neutrophils, simply called neutrophils. Lifespan of neutrophils in the blood is very short. They just live just for 2 days the most. And so that's why we need to produce a lot of neutrophils, 8 million almost per million. Neutrophils, this you should remember as veterinarians. You do white blood cell counts and differential counts. In the case of carnivores, including dogs and cats, there are more neutrophils in the blood than compared to lymphocytes or macrophages, monocytes. Neutrophils are highest in number in the peripheral blood. But whereas in horses, it's only about 50% neutrophils. Rest are lymphocytes, macrophages, eosinophils, and all that. But in ruminants, neutrophil numbers are just 20 to 30%. They have more lymphocytes. Percentage of lymphocytes is almost 50 to 60%. Okay, that's one question that we will see more often. Neutrophils have that multi-lobe nucleus. That's what is shown in this slide. That's all we want to look at. The nucleus has different lobes. But there could be immature neutrophils or the band cells. You see at the most 2 lobes, or sometimes you might just see a band. That's an immature neutrophil. Okay? But neutrophils have at least 3 lobes usually. That's again the same thing, showing different lobes of the nucleus. But importantly, this slide, you should remember there are primary granules. These are the bigger ones, are the primary granules in the cytoplasm, and these are also called lysosomes, as we will see again. Lysosomes, but there are secondary granules. That's very important. Neutrophils have those primary granules called lysosomes, and secondary granules, smaller ones, called just secondary granules. So what's the main function of the leukocyte? That we said already. It's the main first-line phagocytic cell. That's the main important function for neutrophils. But they have other functions. They also produce antimicrobial peptides, sort of antibiotics that can act against the bacterial or viral organisms. But they also play a role in inflammation. That's not very common, but sometimes neutrophils come to the site of inflammation, such as, for example, arthritis, rheumatoid arthritis or something. They come to the site and release their granules to the outside tissue. That's a little bit more damaging to the tissue right there. So that they have some role in inflammation. But their main function is phagocytosis and to some extent antimicrobial peptides. They have these primary granules and secondary granules, which you will see what exactly they are in the next slides. But again, I want you to remember phagocytosis is a 2-step process. The neutrophil has to ingest that bacteria first, ingest the bacteria, and then it has to kill it once it ingests. Some bacteria can evade that killing process. They can live inside the neutrophil or macrophage. But again, remember, phagocytosis is a 2-step process, ingestion first and then killing. And how exactly they kill, we will see in the slides next. But primary granules contain that lysozyme, which we'll talk about when we talk about innate immunity, which can act against the bacteria. And then there are a few other enzymes, myeloperoxidase, elastase, and cathepsin B. All these are kind of enzymes that are degrading to the ingested bacteria. That's how they kill the bacteria, using these. The secondary granules, the first one shown is lactoferrin. We'll talk about this again, but lactoferrin is the one that binds iron from the surrounding environment. It binds to that iron, makes it unavailable to the bacteria. Bacteria in turn need that iron critically for their multiplication. So this lactoferrin binds to that iron and makes it unavailable to the bacteria. Lysozyme again acts against the bacterial surfaces. There are other enzymes that are also degrading to the bacteria. That's what is shown here again, what exactly these primary granules. Again, remember these are also called lysosomes. They contain this myeloperoxidase. Defensins are the antimicrobial peptides that we talked about. There are a few other enzymes and the lysozyme that acts against the bacterial cell walls. Whereas secondary granules has that lysozyme in turn also, but it has that lactoferrin. Lactoferrin, again, is an iron binding protein. It binds to the iron and makes it unavailable for the bacteria. And then there are other enzymes, collagenase, that are also damaging to the bacteria. That's the same slide again, so I will not spend time. Here, neutrophil, we are showing in this diagram that the phagocytosis is a two-step process again. If the red one here is the bacteria, it has been ingested and brought into a cavity which we call as phagolysosome, or simply phagosome first. And then, once the bacteria is in the phagosome of this neutrophil, the lysosomal granules, the primary granule, or the lysosomal granules come and fuse, forming what's known as this phagolysosome. And then these primary granules will release all those enzymes, like lysozyme, myeloperoxidase, elastase, all those, and including later on come the secondary granules that will kill this bacteria or the ingested bacteria. Question?
Is the primary granule the black dot?
Yes.
Okay.
Yeah. The red one is the bacteria that I put, and the white one is the vacuole within the cytoplasm of the neutrophil. That's where it ingests and brings it into that vacuole, and then the primary granules come and fuse, and the whole thing is called phagolysosome. Okay. And the killing process involves release of all those primary and secondary granules. But when we talked about macrophages do the same thing. They bring it, and then they also release some toxic materials. But yesterday we said one specific molecule that they release to kill the ingested organism. It's the nitric oxide. If it is a macrophage bringing this organism, they release nitric oxide also to kill the ingested organism. Okay? But they also have other mechanisms which we'll talk when we talk more about phagocytosis. But that's main function of those primary granules, secondary granules is to kill the ingested organism. But sometimes neutrophils come to the site of inflammation. For example, again, I said, just an example is rheumatoid arthritis. For example, there is arthritis in the joint. Neutrophils come to the site and then release their granules to the outside. And that's kind of damaging to the tissue a little bit. That might lead to erosion of the cartilage and may contribute to that inflammation process to some extent. So that's one thing that's bad about releasing the granules to the outside environment. Okay. We will talk about these immune complex diseases and hypersensitivities in later classes, but remember that neutrophil granules mainly intended for killing the ingested organism, but sometimes they come outside and cause problems. Eosinophils are very small in number, just 2 to 5% or less. But in heavily parasitized animals, animals having a lot of parasites, or even allergies, you will see a lot more eosinophils. Eosinophilia. You know what is philia. Whenever we say eosinophilia or neutrophilia, it's increased numbers. But in parasitized animals and allergies, you will see a lot more of these. Phagocytosis is a function also for eosinophils, that's but very minor. Their major function is to kill the large parasites, bigger parasites that cannot be phagocytosed. They release their granules onto the parasite, and these granules contain these major basic protein is the most important one, but the eosinophil cationic protein, eosinophil neurotoxins, all can be released onto the parasite, and that's how it kills that parasite. As we said, they're also involved. Whenever there is an allergy, you will see also eosinophils coming to the site. They are contributing to reduce the allergies, or they're actually causing more problem there. That's very unclear. Why do they come to that site is another question. Do they think those allergens are parasites? Maybe that's the reason they come to that site and release their granules, causing problems. But that's still a question mark. But their major function is not allergies. Killing the parasites using their granules. And again, don't forget about these granule contents, especially major basic protein. Eosinophils, the nucleus pretty much looks like a neutrophil, but how do we distinguish between neutrophil and a eosinophil? The granules, that's good. They take intense red stain with the eosinophil, eosin, whereas neutrophils take intense bluish stain. And lastly, we are talking about basophils and mast cells. They also have granules, and they also stain intensely with basophilic dyes, but their nucleus is not multi-lobed, so that's distinguishes. But the numbers are very small, less than 0.2% basophils in the blood. Mast cells are mostly in the tissues. There are two kinds, mucosal and connective tissue mast cells, but they both do the same thing. They're mainly intended for acting against the parasites, but they're also involved in allergies, especially type 1, immediate type hypersensitivity, which we will talk a lot in later classes. But how do they act against the parasites? say for example, if there is a roundworm in the intestines of a dog, mast cells do recognize these parasites, come to the site, and then release their granules. Their granules contain histamine. Histamine, how does it act in the intestines? It contracts the smooth muscles. It contracts the smooth muscles, but it dilates the blood vessels. Dilates the blood vessels, so there is a lot of fluid coming into the intestines. And then these smooth muscles are contracting, increased peristalsis, movement of the intestines. So what we call is a self-curing mechanism. That's how animal tries to throw out those parasites to the outside. Okay. So that's how these cells function. But they are involved in allergies. As I said, histamine released into the lung tissue here can cause constriction of the bronchioles and release of a lot of fluid there can cause difficulty in breathing, such as allergic asthma. Okay. So those are the what exactly mast cells do. Platelets derived from the megakaryocytes, which come from the stem cells. Main function, of course, it's involved in what? Clotting, blood clotting, platelets. But they can secrete some factors that